slice of it, and we can see that there's a circuit associated with it. And then we can then look at individual circuits in a rat. Okay, so we wanted to see whether each of these neurons here are connected to each of this neuron, and then whether they could how does this neuron actually process all the inputs so to arrive an output. So the main question that I'll probably show you in this talk is to say that to give you an idea of how many synaptic inputs, or how does the synaptic inputs or how are they organized to arrive in an output. Okay, so so we can look at different regions of this particular neuron. This is a neuron that actually, so this is the surface of the brain. This is the surface of the brain. It goes down 1 millimeter, and there are certain layers, layer 1, layer 2, layer 4, layer 5. And this layer 5 here, this is what these big neurons are in the rat brain, in the rodent brain. And this is what we call a layer 5 pyramidal neuron. It looks like a pyramid. It has basal dendrites because it goes into from the base of this triangle, and it has apical dendrites because it goes from the apex of this triangle. So we're looking at inputs depending on the regions. So basal inputs, proximal oblique inputs, oblique dendrite that goes along this ones here. This is the main apical trunk. And then we're also looking at distal inputs somewhere around here. So how are they organized for this neuron to arrive in an output? That's one of the questions we're trying to answer in this talk. We also wanted to see whether if you have inputs at different regions, different dendrites, how they propagate towards a neuron. And once this neuron fires an output, it actually sends back some information to it back. The signal back propagates to the dendrites and we're now looking at how the back propagation affects the dendrite. And we can actually show that some of these later on in the talk, this back propagation of the input can actually dictate some form of learning process and how and dictate some or say something about how we learn things. All right. So our approach is to reverse engineer the brain. So what we've developed an instrument called optoelectrophysiology. So we still use an electrode, that means the electrophysiology, but we use a lot of optics. So we use optics to stimulate inputs. We use optics to record the activity. We also use neuron modeling, and we also use lasers to cut these dendrites. Another way, our group is also interested in actually building up materials. So we've actually grown neurons on a chip, and we've engineered the chip so that it functions like how our brain works, or the extracellular matrix in the brain, and we can program them to look exactly the same way as neurons in the brain. Because if you just grow neurons on a culture, most likely they don't look like the neurons in our brain. But if you actually engineer the surface of the culturing surface, then you can maybe program the neuron so that it would grow in the same way as neurons grow in our brain. But I'm not giving you some of this talk. I don't think I have time, but I'll probably describe to you this first part here, which is how we try to use optics to understand how our brain works. So I mentioned most of you are probably physicists, so let's just give you some ideas. So each of these neurons in our brain send information to neurons, so other neurons, using synapses. So like this one's here is a synapse from another neuron. Like this neuron here could synapse into this neuron here. There are also negative synapses, these are red ones, and there are positive synapses, these are the blue ones. So the blue one is based from a positive synapse using probably from this layer 5 pyramidal neuron. These are smaller pyramidal neurons would also do a positive synapse to this neuron here. So the typical way for neuroscientists to study that is actually to use electrodes. Okay, this is how a synapse would look like. So this is this sending neuron here. This is this part here, and this is from the receiving neuron. So when the sending neuron sends information to the other neuron via what we call neurotransmitters, this neuron here, as measured from the electrode, will then change in polarity. So this is a transient change. This is about 20 milliseconds in time. This is around 1 millivolt that you can measure from this electrode. So a typical way of for neuroscientists to do that is actually to use electrodes. This is what we call electrophysiology. You can use electrodes at the synapses, and then you can trigger a synaptic input, and then you can also use an electrode at the soma, and then the soma can then measure the inputs from these two electrodes here. However, if you were to use electrodes, you will end up an experimental rig that would look like this. This is about 12 electrodes. This is a very difficult experiment because in order for you to position, this is about, again, as I mentioned, this is about 10 to 20 microns in diameter. The smallest part here is about 1 micron. So this electrode needs to be spatially positioned very accurately towards a neuron. And if your table is not floating, or if it's not isolated with the floor, if you stomp on the floor, then you could actually dislodge the neuron. So this is practically a difficult experiment. If you move the table, these electrodes will actually get out of the cell, and then you'll see something which is not representative of what the neuron is actually doing. So in order for us to emulate synapses, we use optics, and we use light, we use lasers. So we instead of using electrodes here, we use lasers to emulate synaptic inputs. And we only have one electrode that's connected to the soma, which is this one here. And depending on the organization of these inputs, we can then record the output here, depending whether there's single output or a single input or two inputs, and so on and so forth. All right. So how to use light to emulate a synaptic input. The first two methods here, which I've shown, optogenetics and photolysis, are two main techniques used in neuroscience to use light to emulate a synapse. You can, in optogenetics, this protein here, you need to express it you need to genetically modify the animal so that it expresses this kind of protein that would allow, when you shine light in it, it will open up. Okay, so when a synapse actually occurs, channels like this ones open up and allow ions to go inside the cell, and that's why you see a change in the voltage. Positive ions would change the potential difference between the voltage inside the cell and the voltage outside the cell. Okay. So, but then you need to modify your animal to do this. Another way to actually stimulate neurons, or using light to stimulate neurons, is by using a chemical that's already used by the brain, but then you apply a chemical cage. So the gray one here, the gray surrounding here, is actually a chemical cage. If you shine light on it, the chemical cage gets released. The green one is the neurotransmitter. The green one gets released by the cage. It will allow these channels here, which are already present in the brain, to open up. Okay, so this will bind to a receptor and then allow ions to go in. And that would, in principle, emulate a synaptic input. Once you shine light, it will allow ions to go in, and it will change, it will practically emulate a synaptic input as if it were coming from another cell. Oh, there are many other ways to use light to do that, to stimulate cells. You can heat the membrane, and then the membrane becomes porous and allow ions to go in. You can use a heating via a nanoparticle, something like this one's here, and then you can um, uh, and some of the particles would go in towards the cell. You can use you can change the um, magnetic field around the protein, and then allow ions to go into the cell, something like this. If you have this magnetic particle that would have some kind of dipole moment, or you can use photo switching molecules. We've summarized all of these techniques in a review article we published at Journal of Optics. You can have a look at that if you're really interested. So, but what I'll show to you today is the technique we use is photolysis. Okay. So we again we take during an experiment we take a slice of the brain from a rat. So we in the morning we harvest the brain in the morning, and then we slice the brain into thin slices, 300 microns thick. And then this is an example, a picture of a brain slice showing the cortex. The neurons will be sitting around here, and this can be a representative neuron that would probably sit somewhere around here. We then use a microscope and position an electrode into the cell, and then allow a dye to get in to the neuron. And then we use a 2-photon microscope to image the cell. So note that this is an example image of the neuron, but then the neuron will also have different other neurons behind it, because there will be a lot of neurons in here. But we only are looking at this neuron with the dye in it. So once you've seen the dye, you've seen the neuron, and you've imaged the 3D morphology of the neuron, then you can study different areas of neuron, different parts of the dendrite. So um, yeah, so you can see from here, this is a 3D reconstruction of neuron. This here is the electrode. This one here is the electrode. It has still a dye in it, but you can also use electrode to record activity at the soma here. You can still use that. You can either record, you can also use the electrode to stimulate, you can inject current into there, and then the neuron would also respond to that, okay? Once we know that the electrode, we can then produce, we can use light. Again, I mentioned we use photolysis. So these are chemically caged neurotransmitter, and we use what we call 2-photon uncaging. Okay, I won't be explaining 2-photon the process, but the idea is that if we use a highly focused laser, and the absorption of 2 photons by this molecule here allows this neurotransmitter to get released. Okay, so once and the release is just is localized within this focal volume here. So so note that only with neurotransmitters within this focal volume gets released. There's no release somewhere around here and here, and then you can this neurotransmitters then bind to receptors in the neuron, and then allow entry of ions towards the cell. This is just one input. We wanted to see several inputs and something like that. So we wanted to position the laser anywhere within this one, but we also wanted to position the laser simultaneously. Okay, and this is then what's the contribution of physics into this technique. So if we were to produce multiple inputs, then we can then study multiple inputs from different areas of the cell, and then we can then record activity from this electrode here. So how do we produce light patterns, those multiple inputs around the neuron? Okay, So most of you would have seen this movie, Star Wars. This is what holographic projection, where R2D2 has a hologram here and probably have a laser behind. And if you shine a laser onto the hologram, produces holographic projection here, where you have dark spots and bright spots here. And practically, if you change the hologram, you can change the pattern. If you place a neuron along this, then there will be bright spots where some of parts of the neuron would actually respond. There will be dark spots here, something like this one's here, where some parts of the neuron won't respond. So the physics of that is well known, and the holographic projection has been around for several years already. And the idea is that if you shine a coherent light into an aperture, if they are coherent and they would interfere with each other, you can have a constructive interference around here. So you get bright spot, you get destructive interference around here when you have when the beams are actually interfering. If the aperture here is or more complex, something like this, a two-slit experiment, then you have a more complicated pattern, something like this one's here, where you have bright spots and dark spots depending on the organization. If you are able to program or change your aperture here, this is just an amplitude, but then if you have you can actually make an aperture that's consisting of just a face using a programmable hologram, for example, then you can produce arbitrary spots specifically targeted to specific regions in space where you want them to be, right? So this can be constructively interfering there and destructively interfering in these regions here, and so on and so forth. So like this is an actual experiment. This is 1, 2, 3, 4, several spots forming the word ANU. And we can then turn them on and off by just changing your programmable hologram. Okay, you can position the. Of course, this is not Princess Leia, but then this is what we need actually to to um, um, to stimulate our neurons. The good thing with that is actually the neurons would actually be, oh sorry, the spots, you can position them on Z as well, in the Z axis, so you can have full control of the spots in 3 dimensions. So right before I came to neuroscience, I was using the spots here for our what we call optical tweezers, something like this one's here. I mean, I've never, in 2005, I didn't have any idea I'll be working with neurons until I moved to neuroscience in 2007, or to ANU in 2007. So we can then, you can use this individual spots to transfer momentum, like this one here. We can change the position of microparticles, but we can also transfer energy. This is fluorescence from a fluorescent beads. This is in XY, and this is in XZ. If you can, there's a camera at the side, so this is how a point spread function of a focus from coming as viewed from the side. So given the spots, we can then, it's about, we can then use them to position these spots around the neuron if we are able to image the neuron, and then we can program the hologram so that each of the spots would specifically target different areas of the neuron. So this is then what we wanted to do. So so we built this microscope in 2010, 10 years ago, and still is up and running up till now, except that there's no more student at the moment working on it because of the pandemic. But the idea is that we have a laser. We can image the neuron using a 2-photon microscope here. So the cells, or the brain slice, will be sitting here. This is the electrode. This is where we place our cage glutamate, or cage neurotransmitter. And we patch the neuron, and then we image the neuron using this 2-photon microscope, okay? Once we image a neuron, we can then use the holographic projector. So the SLM here is what we call a spatial light modulator, where we can program the hologram or encode the hologram there. This is a digital micromirror device where we can switch on and off each of the spots at high speed. So if we can position already the spots at the dendrite, we can then turn them on and off if we wanted to see synaptic inputs that goes on and off. At, and then all of these, the hologram then is transfers into an optical relay system using these lenses here up to the setup downstairs, where we have this neuron here. The electrode is connected to an amplifier and towards the computer. All right. So to produce a hologram, this is just a basic tutorials. If the hologram looks like this, you can produce light spots that would have two spots along the x-axis. So this is the z-axis here, and this is the x-axis. This is the intensity. And if you have a hologram that looks like this, you can have two spots along the z-axis. This is the x-axis again. This is the z-axis here. If your hologram looks like this, you can have arbitrary positions in the XZ plane. Of course, you can also have in the Y plane as well. So you can have 3D position. So this is just trying to say that if you increase the number of spots by n, the power per each spot is divided by has a 1/n relationship, because practically the power is just divided by conservation of energy. So this is 10 spots, and these are 20 spots. Some of the spots here are out of focus because they're positioned elsewhere along in the volume. All right. We then position those spots into the neuron. So this is a neuron. We have electrode. This is the electrode here. We're looking at this obliques here, which is this one's here, and the basal dendrites, which is this. We're trying to see the interaction, and then we can then try to turn them on and off, and then we can then see that this neuron will fire an action potential whenever it receives some number of input, some threshold. It exceeds some threshold, something like this one here. So this has been published over 7 years ago. So you could see so this is the intensity when the measured intensity when all of the 5 spots are on, then it can turn on, it can produce an action potential. Another experiment by one of my postdocs, if you produce synaptic inputs, this is not cage glutamate, but this is free glutamate. So we produce it out because this is 1 millimeter in distance. So sorry, from the surface here to around here is about 1 millimeter. So this dendrites is about 500 microns to the soma. So it's very difficult to look at that under a microscope. So our field of view in a microscope is somewhere around here. So what we did is actually to produce a glutamate input at the edges of the brain, and then we use our microscope here to look at the interaction between these inputs here and these inputs, and we managed to see some correlation between between the role of these inputs when gating these inputs here. So this is another experiment. We then had some collaborators in Germany where we built the same microscope holographic projector into the existing 2-photon microscope. So this is the setup that we built in Germany. This is their existing 2-photon microscope where they performed experiments. So Veronica is a neuroscientist, and she has a specific question where our system can actually answer. So what we did is that this is an example experiment from Veronica's lab, where we look at the interaction of inputs into a basal dendrite here there are 10 inputs, but no, this is not in the same plane. These are actually 10 inputs that are separated at different XZ planes. So these are different planes here, three different planes, 6 inputs at one plane, 5 inputs in the middle plane, and 2 inputs at the top plane. And each of the input would then produce a corresponding output, something like this one's here. This is an input from a single input here, which is 1. This is the input from 2, and so on and so forth. If we were to produce them one at a time, so this is the output when there's 1, and there's 2 at simultaneously, and there's 3 simultaneously here, and so on and so forth. You can then drive the neuron to fire an action potential. So these are, this is an output when you all have 10 simultaneous inputs into this neuron, making them fire. So what we've seen in this experiment is that these inputs are actually not integrated by the neuron linearly. So if the inputs were integrated linearly, the output response, or the height of these ones here, should follow this line here, because this is just an arithmetic sum. But this is here, what we've measured so far is actually it increases as you increase the number of inputs. So there's some kind of nonlinearity that occurs within the dendrite when there's actually receiving inputs. So it's not a linear integration, just for the neurons to process all of these synaptic inputs simultaneously. Yeah, so that's one of the main findings in this work. So what I've shown so far is that we can stimulate neurons using light. We can position light spots anywhere within a neuron, and then we could measure the response from this electrode. It will be nice if we can also measure here, not just stimulate. We can see how this input is actually propagating towards the soma. So if you were to measure that, we need electrodes. But apparently, we can also use light to measure the response of the neuron. So this neuron here, when they are active, one of the ions that goes into the cell is calcium. So calcium goes into the cell, and if they're active, calcium, the number of calcium inside the cell increases. If you place a dye, a marker inside the cell that reports or senses the amount of calcium, the dye will change in fluorescence because of there's more calcium in there. So it's called what we call calcium imaging. So if we were to measure calcium responses in this one here at this region, we can then look at the change of the fluorescence as a function of time. And this change of the fluorescence as a function of time is correlated with the activity of the neuron. We can position the spots anywhere in different areas, and then we can record activity as a function of time. Okay, this is not stimulation anymore. This is what we call optical recording. So just to give you an overview of what's going on to the cell. Again, this is inside the cell, and this is outside the cell. The boundary between outside and inside the cell is what we call a membrane, and in the membrane there are ion channels. These are ion channels. When the cell, depending on the activity of the cell, certain ions goes into the cell, and it increases the concentration of ions here, particularly calcium. So if you have a calcium indicator, you can then record the changes in the fluorescence of the indicator, and then you can correlate that with the activity of the calcium and eventually the activity of the cell. So this is a cell here. We're just looking at calcium activity. It actually is pulsing. So if you look at the different points in this circuit, then you can see changes in the calcium. because of spontaneous activity. So they're just practically just moving around. They're talking to each other. We don't know what they're doing, but then practically we can monitor what the calcium is actually doing. So this is looking at it at circuits. So a lot of people use calcium imaging to look at brain circuits now in vivo. But again, our question is actually to use calcium to look at individual neurons. And the question we're now looking at, instead of inputs, we're trying to look at how the signal is propagating along the dendrites during an input. So when a neuron when when a neuron sees an input here, it then the parts of here will depolarize. So this is the change in the color, and it will propagate towards the neuron. Eventually, it will fire action potential. So the signal goes through. It will go to different dendrites as well, because I mean there's actually no preferred direction, and then the signal then registers as an action potential. What is really important is that before it reaches the soma, there is what we call a dendritic spike that occurs here. There is some kind of integration that happens at this region before it drives an output here, and this is what we call a dendritic spike. We're then we This is practically important in what we're trying to understand, because I mean this neuron, I mentioned that this is about 1 millimeter or around more than 500 microns in distance from this point to that point. It is very difficult for signals to go through from this point here to here if there was no way of amplifying signals at this region. So in a way, there needs to be some kind of mechanism to relay inputs at the distal ends here so that it reaches the soma. And the main finding for that is what we call a dendritic spike, which was discovered over like 2 decades ago by Matthew Larkham. So this dendritic spike in this region here has been proposed over two decades ago. What then we're trying to what we can also produce that dendritic spike if we actually um, make this neuron fire action potentials. The action potentials then back propagates back towards the neuron, and then it will also drive this part here to fire a dendritic spike, something like. So if we fire action potentials here, this is a modeling experiment. It will then make this neuron fire a dendritic spike that would something like looks like this one here. So if you have 4 action potentials that you make this neuron fire using an electrode, it will produce a dendritic spike also at this region. So perhaps the original question of forward propagating, the effect of these dendrites will actually affect a forward propagating dendritic spike. But then the effect of back propagating action potentials could also be affected by these set of dendrites here. So we are then interested in trying to understand what is the role of these oblique dendrites, whether they are actually syncing information, or whether information is synced through here, or whether information is synced through here when it goes the other way, or whether they actually contribute to the propagation or to the dendritic spike here. And we've actually seen some really interesting results using this, with this question. Can the oblique dendrites, we've actually seen that these oblique dendrites can evoke dendritic spikes. This is the topic of the second part of the talk, where we try to use calcium imaging to look at the dendritic spike at these specific regions of the cell. Okay, so this is a modeling experiment. Again, I mentioned that in 1999, over two decades ago, Matthew Larkham discovered that there is what we call a dendritic spike at this region here, and it is what we call frequency dependent. When you shine, or when you allow the dendrite to fire action potential at 90 hertz, it will not fire a dendritic spike. if it's 100 hertz, it will fire the dendritic spike. So there's some kind of switch. So in a way, our brain would work like there's some kind of frequency of action potentials that practically our brain would actually suddenly um, function, something like that. So what we've what we've seen from our model is that um, this specific set of dendrites that my student Michael was looking at can actually drive the same type of dendritic spike, but at a lower frequency. So this is now 90 hertz, this is 30 hertz, and this is a frequency at this region here. And in fact, this is also a switch where, so here there's nothing that goes on. At 100 hertz, it goes up. Here, what you've seen is that the frequency threshold is much lower. At 30 hertz, there's nothing, but at the 35 hertz, there's something, which is this one here, which is what we call almost a similar type as this one. This is from our model. We then use calcium imaging to verify that. And now we're looking at this specific set of dendrites. And then we then image that set of dendrites using our two-field microscope. These are the dendrites that we see. And then we can produce holographic spots some around well, this one's here. These are the holographic spots that people will position at different areas of the dendrite, and then we can look at the flicker of these dendrites of this fluorescence and correlate that with activity. So from experiment, we've actually seen that these dendrites also produce the dendritic spike that we've seen from our model. So at a certain frequency, there's nothing, but then at a certain frequency here, this is about higher frequency, which is around 70 hertz, you see something. But the idea is that the model, of course, is not representative experiments. So in experiments, we have a higher frequency that we've seen. So this is 70 hertz, and this is around 50 hertz here. Sorry, I forgot to put the label. If you look at this in detail, it will look like this. And again, you see that there's a frequency switch. So if you plot them as a function of frequency, you can see this one here. And apparently, the system doesn't occur in all dendrites. It only occurs in certain dendrites. And that increases the difficulty of experiment because you're looking at the dendrite and you don't know whether it is firing a dendritic spike or not. So there's, again, a change in the calcium here when you have a certain frequency, which is around 70 hertz. We can then plot it as a function of frequency, and this is a sigmoidal plot that represents the step function or the memory function. So this is summarizes our model versus experiments. Our model sees that at certain dendrites, we can see a dendritic spike. At certain, there's different other dendrites, you don't see a dendritic spike. And from calcium imaging, at certain dendrite, we see some response, and at certain dendrite here, we don't see a response. And okay, so what is really interesting is that we can also record what we call an after depolarization at the soma. So this is the recording at the soma. This dendritic spike at the nexus, which was proposed by Matthew Larkham in 1999, can also be seen here as a voltage recording at the soma. Similarly, we can also look at the ADP in the dendritic spikes that we see in these oblique dendrites here. Okay, so so the idea is that we're now looking at ADP because we're just looking, we're not doing any calcium imaging now, because if we can practically just look at the voltage recording at the soma using this electrode here. And that actually makes sense, because if we were to cut the dendrite using a laser, what will happen? So if we use calcium imaging, if we cut the dendrite, then you don't see anything. So, but if we cut the dendrite and we measure the responses of the soma, then we can actually see some kind of change. So so the ADP here, this is from our experiment. You can see that some frequencies, these are different frequencies, it's just aligned to the last action potential. You can see that, again, there's a step function. If you look at that, so at 70 hertz, there is some kind of response, and this is the response from Matthew Larkham's prediction, and this is our prediction here from these ones here. What's going on? Yep. So if we use the ADP to measure the response of the neuron, we can then cut the dendrites. If we cut the dendrites using a laser, then we can then see whether this step function is still there or not there. If and note that not all dendrites would have that kind of response. So we then proceed with a cutting technique, which we actually proposed in 2016. So this is my former PhD student who cut different types of dendrite and see that some of the function of this neuron changes. We then use the same protocol to cut specific dendrites in the regions we're interested in. So if we cut oblique dendrite 3, for example, we then see that there's so if before cutting you have this response. After cutting, we have this response. So if we cut oblique 3, this is the response. That means the dendritic spike is gone. If we cut oblique 1, you still see this step response, which indicates that the dendritic spike does not occur in this dendrite, but it occurs in this dendrite here. Experimentally, we've shown that we cut the dendrites using a laser. So we shine a tightly focused laser. This is about 1 micron in diameter, and then we can then see that there's no more dendrite here. And we can, this is an example, this is from the model, and this is an experiment. So this is equivalent to oblique dendrite 3 in the model. You see that there's no more step response in the ADP, and there's also no step response from the frequency plot of the ADP. If we cut the dendrite that is closer to the soma, you can still see that there's still a response from the ADP, a step response from the ADP. So in a way, we've seen something in the dendrite, that these types of dendrites in the oblique, that they actually have active responses, and they actually produce dendritic spikes. And nobody has seen that before. So what are the implications of these results so far? So if we were to memorize something in a real brain, what do we do? So we need, if you're training how to play a piano, or you're playing an instrument, or learning how to play soccer, for example, you know how to kick, you need to train. And training means you need to repeat everything. If you have a mistake, you repeat again until you practice the whole piece, for example. The same way in artificial brains, artificial neural networks, when you train, you do some iterative process until the weights of your network boils down to or merge or follows a process so that the weights would give you your desired outputs. So it's practically training your network and practically changing the synapses of your network here. In reality, a synapse, again, is the connection between one neuron to the other neuron here. They are connected via synapse. When you're memorizing something, this synapse is actually increased, and this has been proven by the change in the strength of the synapse here increases if you try to memorize. Okay, so practically our memory is stored by the size, by the strength of the synapse. And this has been proven, and this has over 2 decades ago, B and Poo discovered that if this synapse here, if this neuron here is firing and this neuron here fires after when this one is firing, then they are correlated. The strength of the synapse increases. That means this synapse here, so when this one is firing, before this one is firing, this one this one here is leads this neuron to fire, drives this neuron to fire an output. Okay, so when this one here occurs after this one, and it's not correlated. Okay, so this one here is when it's not correlated, but this one here is when it's correlated. So that means the synapse is actually increasing. So and what do we mean by a synapse that's increasing? So this is what we call long-term potentiation, a typical memory theory in neuroscience. So the signal that you measured in this neuron here, when you have a bigger synapse, is actually a larger signal. A larger signal, that means physically the connection between the two neurons are actually large. So this is a smaller connection. There's ion channels that are smaller, but then you have a larger connection, you get more ion channels, you get larger area, and you get a larger signal that's actually represented. Okay, so this neuron, this set of memory is actually stored something like that. There's many synapses all over, and the size, the change in the synapses forms our memories. So, what are the implications? What is the role of the dendritic spike in plasticity? Okay, so if this neuron here fires an action potential, it drives this neuron. Note that when during our experiments, one input here is just a small blip. This is just a small blip somewhere around here. If you measure this when this one is firing, what you see here is just a small blip. And this is because there's no extra boost from a dendritic spike. Okay. If note that in fact, experimentally, I've shown this earlier, that each of the synapse will just give you this one, and you need at least 10 of those to drive this neuron to fire an action potential. So in principle, the theory of B and Poo is that if you're looking only at two neurons driving this neuron, it will be really difficult for this neuron to fire an action potential if there's nothing going on within this neuron. So what if there is a dendritic spike that occurs here simultaneously during a spike, then the probability of this neuron firing an action potential will be higher, because this part here is already depolarized. So if this one is depolarized and this one is firing an action potential, then this neuron will have higher probability of firing an action potential. So this theory here is well known in these regions because this is well studied. This is closer to the surface, and this is a lot of people are studying the apical tufts, but actually nobody is studying this region here, which is the obliques. And in fact, if you look at a typical tree in the forest, for example, like this one's here, which I took a couple of days ago, this is just the apical trunk of a pine tree. The oblique dendrites are actually known to be old dendrites that are outgrown because, I mean, there's no more sun here, so that they receive no inputs, so there's no leaves. They're like dead dendrites, are practically outgrown dendrites as well. You don't see them very often. You don't see them functioning. Nobody's really interested in it. A lot of people are interested in the apical tufts because there's more activity there. There's a lot of leaves there, and there's more inputs. So what we've seen so far is that these oblique dendrites are not passive. They're not outgrown dendrites. They actually have active properties. They can actually have dendritic spikes. And if they have dendritic spikes, they can actually store memories in terms of synapses. So these dendrites here are really important. It's not just these dendrites here. And that's what the recent study is all about. Okay. So in summary, I've shown you a technique called holographic projection, where we try to use them to stimulate neurons in the brain. We can stimulate the neurons, and we can also use the light beams for calcium imaging. And then we also analyze the propagation of dendritic spikes along the dendrites of these neurons. All right. To end, this is my group. All of them actually moved on already. Michael, my former student, has finished his PhD, and did the first part of the experiment. Is a postdoc at Imperial College. We will have some collaborators at University of Regensburg, and this is the funding I've received from the Australian national government. Thank you very much for your attention.
Thank you, sir.
Thank you, sir. We thank you, sir, for the amazing presentation. You have picturesquely explained the fascinating aspects pertaining to neuron stimulation, the computing power of neurons, and shed light on holograms and two-photon microscopy. The lecture would have certainly illuminated our participants. Now the session is open for questions. Participants can put forward your questions in the chat box, or you can raise your hands through reactions. Participants, if you have questions, please raise your hands. You can unmute your mic and then ask the question.
Sir, I'm Justin from NIT Trichy. I have one question regarding this memory aspect.
Yep.
So memory improves through training, as you told, right? So, like, no, with the aging, will it deteriorate, or we can improve the performance?
Memory is stored in terms of synapses. So aging degrades some of our neuronal function, and that also means degrading some of those synaptic connections. So in a way, when we age, if it's natural aging without any disease like Alzheimer's, eventually cellular function will also be degrading, and that's practically very common. What is a problem is when you have disease called Alzheimer's disease, and that would expedite the degradation of the neurons and as well as the synapses, and that would also be in principle affect memory.
Thank you.
Participants, please enter your questions in the chat box, or you can unmute and ask.
I'm Jaidevan from Japan. In one of your slides, you were showing about the usage of nanoparticles, if I remember correct. What is the type of particles you use?
So we've only done one experiment with particles, and that was in collaboration with my colleagues from engineering, and they've used a titanium functionalized titanium nitride particles, I think. So, but what we did is actually to functionalize them with lysine, and we allow them to be sensitive to one of the proteins. That was work done several years ago, and we haven't really done much more on that. The review that I've written in 2017 had more particles mentioned. Those are work from other people.
Thank you.
Are there any more questions? So there's a question in chat box. Which advanced material is suitable to make the fins equipment in IC engines?
Hey, Naren, comedy.
So I'm not too familiar with IC engines. Integrated, what and what we've done so far in terms of using advanced materials or meta surfaces to grow neurons on was actually to grow on certain nanowires. We can have silicon nanowires as well, and then um, and we can grow neurons in them, and we can change silicon, the spacing of silicon of the nanowire, so that we could program the growth of the neurons. Does that answer your question? I'm not familiar with IC engines though.
There's one other question, sir. The transfer of signals are random in brain. So while stimulating them using computer, is the memory stored in a random manner or is it structured?
Yes, that's a good question. So that's the signals that our neurons brain receive. We think of it as random because we don't know what's going on. But from my hypothesis is that there's probably a correlation to whatever input you have, or whatever. So whenever a neuron receives inputs, it will receive from multiple locations, from your memory, from your senses, from your eyes, from your nose, and stuff like that. And all of those are integrated into neuron. Whether that set of inputs are random or they're actually correlated to your synaptic inputs, that's still a big question. Because, I mean, people will just say it's random because we don't know what's going on. But if we know what's going on, then they're not probably random. They're probably specific sequences that the neuron actually receive, that forms that sets a neuron to fire a certain output depending on the succession of this, what we sort of random inputs. So that's one of the biggest questions that we're probably looking at. Is it a random or is it structured? And that's why we try to emulate these inputs in a structured manner to see whether we could emulate whatever output the neuron is actually producing.
Okay, sir. There's another question. What is the prerequisite for doing this type of research? They're asking what type of subjects lead to this research.
Oh, that's a very good question as well. I'm a physicist, but I didn't know anything about neurons until 10 years ago. I have done a lot of optics. And I didn't even know whether I would go to neurons, but before coming to the Australian National University. So I was just driven by my curiosity, because I have this technique which we developed over 10 years ago, and then we wanted to find applications, and that brought me to neuroscience. And when I was in neuroscience, I was a postdoc, but then I was actually studying everything from zero. I studied a lot of neural activity, how the neuron works. So neuroscience is a multidisciplinary field, and it requires a lot of experts. So if you become an expert in one field, eventually you could probably find some connection with some questions in neuroscience. So my postdoc that came, Vinny Gautam, she's from India. She finished her PhD at Delhi, at IIT in Delhi. She has a degree in physics, and she was doing material science. So when she came to my lab, I actually asked her to build a suitable biomaterial for our neurons to grow on, and that's where we found out how nanowires could actually change the structure of the neurons that we're growing, rather than just growing them on a culture. So in principle, she has a different expertise, but then you can find an application. So my advice is that for you to find your own expertise depending on your strengths and your skills, depending on your field. Don't try to study too many things at the same time. Otherwise, you won't be an expert in one field. So if you are an expert in something, then you can diversify later on to find applications. When you collaborate with other people, when you go to conferences and study different fields, and then you can, in principle, apply your expertise to certain problems. I hope that answers your question.
Yes, sir. We'll take up one last question. The question is, how are the dendrites and synapses are affected in schizophrenia paranoia patients? Can we study the brain using this technique and possibly find treatment?
Yeah. So that's a really nice question. So what we've studied so far are neurons from a healthy brain. So we're trying to understand. So we're looking at the fundamental process of individual neurons. So right now, there are other different topics, areas in neuroscience that model animals. So they make genetically change the makeup of certain animals so that they would behave like schizophrenia. They're bipolar, or some models would actually make some animals have degenerative disease like Alzheimer's. So there are geneticists. So the geneticists then try to produce some animal models, and then we can then use those neurons in our study. We haven't done that so far, but because we're practically interested in the fundamental aspect. But in principle, the understanding of how these neurons would actually be affected in terms of function when the neuron is coming from a schizophrenic mice model compared to a wild type or a natural or a healthy mice model or a rat model, then we could then study their effects.
We thank you very much, sir, for your valuable time with us. Thank you for joining today in IWAMA 2021.
Thank you very much as well. And I'm excited with your questions. Thank you.
We shall move on to the second talk of the day. I shall have the honor of introducing our next speaker, Professor Dr. B. Jayadevan. Professor Jayadevan obtained his PhD from Tohoku University, Japan in the year 1994. He has versatile experience as a researcher, research associate, and lecturer between 1994 and 1999 at CISIR Sri Lanka, his alma mater Tohoku University and Akita University. After working as an associate professor at Tohoku University till 2006, Dr. Jayadevan became a professor at Tohoku University. Currently, he is a professor and the chair of the Department of Material Science at the University of Shiga Prefecture, Japan. His main fields of research include synthesis of magnetic, conducting, photocatalytic nanostructures, polyol and alcohol-based techniques to synthesize size and morphology-controlled nanostructures. He collaborates avidly with several universities across Japan, Daytred University, Paris, NIT Tiruchirappalli, India, and Institute of Material Science, Madrid. He has about 230 publications with over 6,600 citations, and he has authored 5 books. He is also the steering committee member of the Japan Society of Magnetic Fluid Research. With high esteem, I now request Professor Dr. B. Jayadevan to deliver a lecture on present status and prospects of polyol alcohol-based techniques for the synthesis of bimetallic nanostructures. Over to you, sir. Please share your presentation.
Thank you.